In this lesson, we'll continue our investigation into the principles of development. We've already learned about the great puzzle of development, and that is, how can cells with the same chromosomes become different types of cells? We would say they specialize into skin cells and nerve cells and blood cells. They all have the same chromosomes, which means they all have the same set of genes, yet they become different. Well, we answered that puzzle by saying that the immature blood cell is going to be turning on the blood genes, and so the cell will make hemoglobin proteins. The immature muscle cell will be turning on actin and myosin genes, and so they will be making those proteins. Here we see clearly the blood cells and muscle cells look very different. They look different because they're making different proteins. They're making different proteins because they've turned on different genes. If we took a look inside the nucleus of the immature red blood cell, we would see this scene here. The hemoglobin gene has been turned on. And remember what it means for a gene to be turned on. That region of DNA, the two strands separate so that a messenger RNA molecule can be built using the template strand of DNA down here. We don't see the protein doing this. The messenger RNA will detach and go out into the cytoplasm to the ribosome, and then the protein will be built. So the hemoglobin gene is turned on here, and notice over here in the corner the actin and myosin genes are wrapped up. So those genes are turned off. They're not accessible. So messenger RNA cannot be made from that region of DNA if the DNA is wrapped up. Now that's what's happening in the immature red blood cell. In the immature muscle cell, the picture is the reverse. So the actin and myosin genes are accessible, right? They're unwrapped. And here we see the DNA strand separating, and messenger RNA will be built from the template strand for both of these genes, and the hemoglobin gene is wrapped up, turned off. So the explanation at this point is that cells specialize when certain genes get turned on and others remain off. And that different pattern of gene activity explains why cells end up making different kinds of proteins. In this lesson, we'll ask the deeper question. The next question then is, what is it that is turning on the hemoglobin gene in this cell, and what's turning on the actin and myosin genes in this cell, right? What's controlling the pattern of gene activity in different cells? And to answer this, we're going to make a distinction. We're going to use an analogy. And this is an analogy from the realm of economics. And some economists in the past would classify different kinds of jobs in the economy as blue-collar jobs or white-collar jobs. Now, white-collar jobs tended to be sort of the managerial type of jobs, and the blue-collar jobs where you're kind of working with your hands and doing stuff and building stuff. And we're going to use this analogy to understand genes. We're going to assign some genes the label blue-collar genes. Now, blue-collar genes are making proteins, myosin and hemoglobin, and these are blue-collar proteins because they do their job outside of the nucleus, in the cytoplasm or the membrane. So myosin and actin and hemoglobin would all be blue-collar proteins, so their genes would be blue-collar genes. The blue-collar proteins work in the cytoplasm. If we think of a cell, the nucleus is going to be the office, and the cytoplasm will be the factory. Blue-collar proteins work out in the cytoplasm or on the membrane. Now that's going to be in contrast to the white-collar genes that will be working in the office. White-collar genes make proteins, white-collar proteins, and their job is to work inside the nucleus by interacting with DNA. So it's these white-collar proteins that will control which genes are going to be turned on and off. So again, the white-collar proteins are doing their work in the nucleus of the cell by interacting with DNA. Now remember, all proteins are built in the cytoplasm. The ribosomes are located in the cytoplasm. Blue-collar proteins will stay in the cytoplasm or go to the membrane to do their job, but the white-collar proteins are going to go back into the office, the nucleus, to interact with DNA, to control the genes in the nucleus. Now, how do white-collar proteins actually do their job? Well, this picture captures a bit of what's going on. Now, it looks complicated, but let's just take a look, take it one step at a time. Here we see the DNA molecule. And apparently here we have the coding region of a gene, so this would be the recipe for some protein. And if the cell's going to make this protein, it will have to unzip the DNA, separate the DNA strands, and then use one of the DNA strands, the template strand, to build messenger RNA. And when that's happening, we say the gene is turned on. Now, the protein that builds messenger RNA is RNA polymerase, and here we see it sitting right here in front of the coding region of the gene. So this protein's job is to make the messenger RNA. But the white collar proteins are pictured here, the blue and the green and the red and the green up here. So these are proteins that can stick to certain regions of DNA near where RNA polymerase binds to DNA, and they can either help RNA polymerase find its binding site or block RNA polymerase. See, so if these two proteins, because they stick to this DNA region here, if they are helping RNA polymerase do its job, then these two proteins, the blue and the green one here, we would say they are helping to turn on the gene. And this is the way that a white collar protein can operate. It can help express a gene by helping RNA polymerase do its job. Now, it turns out there are white collar proteins that can bind to DNA far away from the actual gene. These are called enhancer sequences of DNA, and proteins can bind here, and they too can actually help RNA polymerase find its binding site and do its job. Even though they're so far away on the DNA, remember the DNA sometimes can coil up, so these proteins can actually stick to these proteins, and this all provides a larger binding site for RNA polymerase, and that will help turn on the gene. So we don't want to get too complicated here, but the basic story here is that white collar proteins can help turn a gene on. And they do this physically by binding to DNA near a gene and helping RNA polymerase do its job. Of course, the flip side of that is these white collar proteins might interfere with RNA polymerase doing its job, and so those white collar proteins would be turning off a gene. So there are different kinds of white collar proteins, but notice their job is to control the expression of a gene, whether the gene is going to be turned on or turned off. So back to our cells specializing into blood cells and muscle cells, we want to understand then that it's the white collar proteins that are turning on the blue collar genes. So when we ask the question, well, what turned on the hemoglobin gene? It was a white collar protein. So why is the hemoglobin gene turned on here? Because white collar proteins helped turn it on. Why are the actin and myosin genes turned on over here? White collar proteins helped to turn on those genes. And we'll see there are other white collar proteins that are turning off genes. The hemoglobin gene is turned off in the muscle cell. The actin and myosin genes are turned off in the blood cells. In this picture then we sort of see the general idea of what white collar proteins can do. Here we see a stretch of DNA. We've got two genes here and one here. And this one here makes the blue protein, and this one here makes the red protein. These are white collar genes that are making white collar proteins, and we know they're white collar because the proteins are interacting with DNA. The red one here is going to help to turn on the hemoglobin gene, and the blue one here is going to be helping to wrap up the actin and myosin gene. So what we have here is a picture in the immature red blood cell. So this cell will specialize into a blood cell because the hemoglobin gene has been turned on and the actin and myosin genes, those are muscle genes, have been turned off. But what we're seeing here is the operation of white collar proteins. Some white collar proteins can turn genes off, for example, by wrapping up, and other white collar genes can turn genes on, binding to DNA and helping RNA polymerase do its job. Now let's take a step back again and take a look at our diagram of early development. Here we have the fertilized egg going through rounds of the cell cycle, mitosis, producing more and more cells. Now what's important to understand here though is that early in development, as we see down here in these pictures, the cells all look alike. So they have not yet specialized. Nevertheless, something important is happening in the nuclei of all of these cells. So what we want to think about then is during early development, these cells here we're going to imagine inside the nucleus of all these cells, white collar genes are being turned on. And then the white collar proteins are going to be doing their work in the nucleus, binding to DNA and turning certain genes on and off. And that's going to be causing the blue collar genes to be turned on and some turned off. And blue collar genes make blue collar proteins, and that's when the cells will begin to specialize. So here we see red blood cells and muscle cells. Of course, they look very different from each other, and they look different from early embryo cells. The early embryo cells all look alike because they have not yet turned on blue collar genes. However, the white collar genes have been turned on. It's just that because the white collar genes and the proteins do their work in the office, in the nucleus, they have not yet begun to specialize. They don't look different at this stage. Nevertheless, the white collar genes will make white collar proteins, and they will operate and turn on blue collar genes, and that's what makes the cells specialize. Now, at this stage, then, we can ask the question, well, what's controlling the white collar genes? What's turning on white collar genes? After all, if you look at this diagram, you'll notice that this cell here and this cell here are going to have different futures. Right? They have different descendants. This cell is the ancestor of the muscle, blood, nerve, and skin cells, while this cell is the ancestor of liver cells, intestine cells, and the sex cells. Well, why is that? How is it that these two cells are going to have different futures? Well, we need to ask the question then, what's sort of controlling white collar genes? Evidently, there are different white collar genes being turned on here because they're going to have different destinies. The answer for that, then, keeping with our analogy, is going to be a CEO gene or several CEO genes. These are genes whose protein product is going to be another switch. So it is a white collar protein, but it's going to turn on and off other white collar genes. We're going to call it a CEO gene because it's responsible for controlling the expression of other white collar genes. We can think of these white collar genes as middle management now, with the CEO being top management. And then again, with the white collar genes making white collar proteins that will turn on the blue collar genes. So what we have here is a hierarchy of management. We have CEO genes making CEO proteins. The CEO proteins are going to control the middle management white collar genes, and those white collar proteins will control blue collar genes. Now these CEO genes, of course, are going to be operating early in development, all the way back to the fertilized egg. So again, we want to understand how could, even at the two-cell stage, how could these cells have different destinies, right? Give rise to different types of specialized cells. Well, let's take it all the way back to the fertilized egg. Here we see a sea urchin cell going through the first mitosis. So it's going to split into two cells. And our puzzle is going to be, how is it that these two daughter cells are going to have different futures? There must be something that's already different between these two cells. Well, imagine if we could look inside the fertilized egg. Here we see the cell captured in the process of the separation stage of mitosis. We got the chromosomes going to opposite sides. Imagine already in the cytoplasm of the fertilized egg is a very powerful protein. Right, so this was already in the unfertilized egg, for example, and then after fertilization, this protein gets localized to one side of the cell. And if the plane of division is right here down the middle here, what we have then is one daughter cell will get the CEO protein, the other daughter cell will not get the CEO protein. That has important implications because the CEO protein, remember, is going to be switching on a certain set of white collar genes. And the idea is, even though there's no CEO protein pictured over here, there would be other proteins that would be turning on a different set of white collar genes here. See, so already at the two-cell stage, we can have different futures for these two cells because the CEO protein was localized, which means that the pattern of gene activity for this cell will be different than the pattern of gene activity for this cell. So, to complete our story then, we've got blue collar genes are making cells specialized. They were turned on by white collar genes, and white collar genes were controlled by CEO genes. The CEO protein was already present in the fertilized egg, located in a sort of uneven distribution in the cells, so that the first division means that one cell gets some of the CEO protein, the other cell does not. That means different white collar genes will be turned on, and so we get different developmental futures for these two cells. In summary, then, we have white-collar genes orchestrating a complex sequence of gene activation to make cells specialize in different ways. And so this is the secret to animal development. It is the managerial genes that are shaping the destinies of cells. Cells specialize in different ways because they're having different sets of genes turned on and off. And the result is a body composed of specialized cells all cooperating to keep the organism alive.